All right, I hope that you can see my slides as they should be, right? Very good. Okay, so yeah, thank you very much to the organizers for putting together this very stimulating workshop. And so like in some of the other talks, in my talk, there will be no phylogeny, but there will be a little bit of inference, and there will be lots of models to data and back. And I guess in this audience, I don't really have to motivate this talk very much. We all know that antibiotic resistance evolution is on the one hand an important medical problem, but on the other hand also a very nice and in many ways interesting model system for evolutionary questions. And so what I would like to try to do today is to discuss two recent case studies that we have been involved in, involving on the one hand the problem of repeatability of resistance evolution against beta-lactam antibiotics, and on the other hand a recent study on the concentration-dependent evolution of ciprofloxacin resistance. And in both these cases there was a collaboration with an experimental lab. In the first case, this is the lab of Ariane de Visser, with whom we have a long-standing collaboration, and in the other case there was important contributions from Bartek, Václav, and Rosalind Allen. So let me start with the first story. So TEM1 beta-lactamase is an enzyme that breaks down beta-lactam antibiotics. And this is, of course, a famous molecule that appeared in previous talks, yesterday in Martin Weich's talk and in Dan Weinreich's talk, among others. So these beta-lactam antibiotics, the most famous representative of which is penicillin, target cell wall synthesis, and they are broken down by these beta-lactamases. And this is a particular version called TEM1. And this has become, over the last years, a model system for evolutionary studies. And typically what one does is that one exposes this enzyme to a novel antibiotic that it has weak activity against, and then one can see how the enzyme adapts to that. And that then gives rise to a kind of resistance landscape of the enzyme. And this, by now, iconic graph of a piece of the TEM1 resistance landscape appeared already in Dan's talk yesterday, and he is, of course, the author of this seminal work. So briefly, what we're seeing here is a subset of multiple mutants, combinations of 5 mutations that when they occur together, which is on this side, so this is the 5-fold mutant and this is the wild type, increase the resistance to a particular antibiotic, cefotaxime, by a factor of about 40,000. And these others are sort of a subset of the intermediates, and the key point of this paper, and I would like to ask you to remember this because I will come back to that towards the end of the talk. The key point of this paper was that if you ask how many pathways there are for evolution to go from this wild type to the 5-fold mutant, only a small fraction of those are monotonically increasing in resistance, a property that Dan and collaborators called accessibility. And altogether, in this case, there are 120 mutational pathways because there are 5 mutations which can occur in any order. Now, this is, of course, only a small piece of the resistance landscape of TEM1, and we have, in joint work with Ariane, over the years explored various other parts of that landscape. So this is a plot from a 2013 paper. And this shows now 4 or 3 sort of small scale landscapes. This is the Dan Weinreich's landscape again, or rather a piece of it, because one of the 5 mutations was omitted here. So these are landscapes consisting of 4 mutations. What is plotted here is always the resistance as a function of the number of mutations. So here you have the single mutants, the double mutants, the triple mutant, and the single 4-fold mutant. And these 2 landscapes, A and B, they were constructed by putting together mutations that were individually particularly beneficial for the resistance. Okay, so basically here we were looking at the landscape in directions where it seemed to go up rather steeply, Whereas in Dan's case, he was looking at this landscape in a kind of retrospective way. He already knew that there was a highly fit mutant, and he sort of reconstructed the landscape with hindsight. And what I just want to point out here is that these landscapes look rather different. So these sort of go up a little bit, but then they go down again, which means that if you start combining individually beneficial mutations, very soon they start becoming deleterious. So there's a strong sign epistasis, which is not really seen in the Weinreich landscape. So here we're sort of looking at this TEM1 landscape in a couple of different directions. This is, of course, still a very restricted view of the landscape because the enzyme altogether has about 2,500 possible point mutations. So, you know, there's an enormous space of things that could happen. So the more recent project that I want to describe to you was actually an attempt to look at this problem of beta-lactam resistance evolution on a larger scale. And so these were experiments that were started by Martin Schrenk, a postdoc in Arjan's lab already quite some time ago. And so here the idea was to actually look at cefotaxime resistance in E. coli in vivo, that is in sort of real life evolution experiments with E. coli, okay? And these were the bacteria that had the TEM1 gene. So there was a highly expressed TEM1 gene on the plasmid. So in principle, they could adapt to cefotaxime by generating mutations in TEM1, and some of them did that. But of course, they could also do other things. So they had the whole chromosome available to them to generate mutations. And the way the experiment was done was by gradually increasing the level of cefotaxime in such a way that the cell density remained approximately constant. The experiment ran over 500 generations, and we were particularly interested here in the effect of population size. So there were two different population sizes, small populations of 2 times 10 to the 6 bacteria and large populations of 2 times 10 to the 8. And so these approximately 100 lines were evolved for about 500 generations. And then if you sort of look at the end what has happened, so if you sequence the endpoint clones, we discovered that there were about 1,200 mutations, both in the plasmid and the chromosomes, and they were could be divided into classes. So there are about 700 point mutations, 267 relatively small scale insertions and deletions, and about a bit more than 200 large scale duplications and deletions. And you'll see in a moment why I make this distinction. So as I said, these mutations occurred both in the chromosome and in the plasmid. And the number of mutations per line was approximately, that had fixed per line, was approximately 10 for both the small and the large population. So in fact, despite the fact that the mutation supply in the large populations was much bigger, the number of fixed mutations didn't differ very much, which already indicated that there was a lot of clonal interference going on in these populations, as you might expect. And so the primary interest here was to look at the repeatability. So we basically looked at pairwise similarity between the mutations in different clones. And the first pattern that we saw was sort of what you would expect. So this is a measure of pairwise similarity. This shows sort of four columns. On the one hand, large and small populations on the level of the nucleotide, and then large and small populations on the level of the gene. So you see that there is a bit more repeatability in the large populations, as you might expect, because you would expect that selection is stronger in the large populations. And there is also significantly higher repeatability on the gene versus the nucleotide level, which is again something that you would expect and that other people have seen in other systems. However, a very surprising at first pattern arose when we looked at the repeatability separately in the different mutation classes. Okay, so this is now pairwise similarity distinguished between the single nucleotide polymorphisms and these structural variants. And here you can see that repeatability is driven in the large populations by the SNPs, but in the small populations is driven by these large scale mutational events. And so there seems to be a shift in the mutations that these populations mainly use and that are sort of preferentially selected as you go from the small to the large populations. And the hypothesis that we developed was that because of clonal interference, large populations tend to adapt via mutations of large effect, which then, of course, typically have rather small rates, whereas these small populations are dominated by these structural variants, which have high rate but small effects. Now, how can you sort of test or confirm this hypothesis? So we have basically two sets of arguments. On the one hand, of course, you can sort of look at how things happen in time. So for that purpose, Martin conducted or sequenced 5 small and 5 large populations at intermediate time points. And from that, Mark Swart then reconstructed these Muller plots. But I should of course, these are sort of to some extent an artist's impression, because the only information that we actually have are the genomic content of the population at these different intermediate time points. But nevertheless, you can see a clear pattern. So what you can see is that in small populations, these red and green events tend to happen first, which are in our color coding, the large scale duplications and deletions, whereas in the large populations, the blue events occur first, and these are the single nucleotide polymorphism. So that already tells you that there seems to be a higher supply of SNPs in the large populations and a higher supply of duplications and deletions in the small populations. Now, the other thing that we did, and this is where the problem of inference comes in, and this was work by my postdoc Sungmin Wang, was to try to infer the selection coefficients and the mutation rates of these different classes. And what Sungmin did was to run Wright-Fisher simulations with three classes of mutations. And so essentially the model then has 6 parameters, namely the rates and effect sizes of these 3 classes of mutations. And this and the data that we use to determine those were the numbers of the different classes at the endpoints for these approximately 100 different populations. Okay, so the problem is in some sense to learn the functional relation that would give you the observed numbers, or rather their mean and variances at the endpoint as a function of these parameters. And so if you do that, you find a clear hierarchy. So you see that indeed the selection coefficients for the SNPs are larger than those of the indels and larger than those of the CNVs. And also on the level of the mutation rates, we see that the SNP mutation rates differ from the CNV mutation rates by a factor of several hundred. Now, I think the absolute values of these, for various reasons, the absolute values of these parameters probably shouldn't be taken too seriously, but I think the relative values are informative, and in particular, the ratios of the selection coefficients are consistent with a generalized linear model analysis on the MIC values, where you basically just try to estimate the contributions of the different mutations to the MIC. Okay, so that's essentially the first story that I wanted to tell you, and I should say that this work is available as a preprint on bioRxiv, which was posted just a few days ago. Now, this model that we use to describe this experiment is sort of oversimplified in several respects. And for one thing, as I mentioned in the beginning, these experiments are actually conducted at changing antibiotic concentrations. And also the model completely ignores epistatic interactions between different mutations. And this sort of brings me to the second story, which addresses exactly those two things, but for a different system. That is, it addresses on the one hand how these resistance landscapes change with concentration, and on the other hand, what the role of epistasis, what kind of epistasis you can expect. And this is, as I mentioned in the beginning, joint work with the Edinburgh group, and the lead author here was Suman Das, who is now a postdoc in my group, and this was published in eLife last summer. And so here the key term is adaptational trade-offs. So to explain that, I need to say a little bit more about how one typically quantifies antibiotic resistance. So the standard way of quantifying antibiotic resistance is through dose response curves. So basically what you do is you measure the growth rate of your bacterial population as a function of antibiotic concentration, and this will typically go down. And what you see in this panel here are two dose response curves, the blue one for the initial, let's say, susceptible or wild type strain, and the red curve is the corresponding curve for a mutant. And so what you see, and this is very common, you see a kind of cost of resistance, that is, at zero concentration, the mutant grows more slowly, but of course it can survive to much higher antibiotic concentrations, right? So generically, and you know, this is not true for all mutations, but many mutations that increase resistance at the same time decrease growth rate in the absence of the antibiotic. And this has the important consequence that the dose response curves of the mutant and the wild type will typically cross. So you have one regime where the wild type is fitter, and you have another regime, which is a so-called mutant selective window, where the mutant has higher fitness. So this is, of course, well known. And what we were interested in exploring in this project was what happens if you now have multiple resistance mutations. In particular, if you have multiple resistance mutations and their combinations, then of course each mutant will have its own dose response curves, and these mutants will cross. And as a consequence, this will induce, as I will show you, a context dependence, that is an epistasis between these mutations, which depends in a certain way on the concentration of the antibiotic. Now this, of course, per se looks like a very complicated problem, because you have this combinatorially large number of mutants, and for each mutant you would have to determine the dose response curves. But it turns out that this problem actually can be simplified significantly, and this is where the experimental data from the Edinburgh group comes in. So it turns out that they measured a bunch of single and double mutants in E. coli conferring resistance against ciprofloxacin. So this is now a different antibiotic. And so here you see in a kind of binary notation these single and double mutants, and you see these various dose response curves, and you see that in most cases there is this kind of trade-off that I discussed, that is, the resistant mutants have lower growth rate and higher resistance, okay? Now this, as I said, this looks very complicated, but it turns out that it's actually not that complicated, and you can actually scale these data essentially to a single curve, right? So if you, and this is of course not something that a physicist immediately would do when you see these kinds of data. You scale the growth rate by the growth rate at zero concentration, and you scale the resistance, you scale the concentration by the resistance concentration, and then you get this rather nice data collapse. And in this particular case, this curve, which is the dashed line, looks approximately like a hill function, but this is not very important at this point. So the important thing is that these curves, to some extent, all have the same shape. Okay, so this means that at least the shape of these curves is not something that you have to determine separately. Now, the second helpful observation is, with this, essentially, once you have this sort of universal curve, you only need to know the growth rate at zero fitness, and you need to know some measure of resistance. So these are now two, if you will, phenotypes that characterize this dose response curve. And so the second observation is that these phenotypes, which we call the marginal phenotypes, are largely non-epistatic. That is, they can be, to a good approximation, be combined multiplicatively. That is, if one mutant decreases the growth rate by a certain factor and another mutant decreases the growth rate by a different factor, then the double mutant will, to a good approximation, decrease it by the product of these two factors, and the same applies to the resistance. So this is shown in this table, which you probably can't read, but the main thing is that the gray parts of this table show comparisons between the non-epistatic predicted null fitness and MIC in cases where this comparison is within the error bars correct, and the purple ones show deviations. Turns out that the deviations all go in one direction, which is in the direction of negative epistasis. And in a certain sense, as I will mention later, this actually still is included in our model. And this pattern, I think at this point, we don't have a good understanding of when this is true. It's true for this particular data set for ciprofloxacin. It has also been seen by Dan Anderson and his group in other systems, but I think at this point it still needs to be clarified under what conditions this is actually the case. But what we did now is to say, okay, let's assume that this is actually true, and then we can build a model which is actually rather simple. And so the model looks like this. So we assume that there's a certain number of resistance mutations, L. Each of them has a null fitness and a resistance value, and these are defined relative to the wild type. So mutant I decreases null fitness by Ri and increases resistance by Mi. And then we can, if we now characterize a mutant by a binary string of length L, then we can get its dose response curve by the scaled version of this sort of universal dose response curve, which is independent of the particular genotype. And it only has two parameters, namely the null fitness in front and the resistance, and these combine multiplicatively. Okay, so this just means that our sigma is a product of the R's of the mutations that are included in that particular multiple mutant. Okay, so that's the model. So here, if you now scale this or choose this curve in such a way that f of 1 is equal to 1/2, then this simply means that these m values simply quantify the concentration at which the growth drops by 50%. Okay, and so this is a huge simplification because it means that we can basically predict or compute the concentration-dependent fitness of all the mutants from only L single growth rates and L resistance values. And so this is now going from the data to the model. So now the question is, what does this model predict? And just to sort of explain this in a simple case, if we only have two resistance mutations, and here we have, let's say, this is the wild type dose response curve. This is one single mutant. This is the another single mutant, and this is the double mutant. Then you see that you will have these crossing points, and at each crossing point, the fitness rank between the corresponding mutants will change, and this you can sort of depict schematically in a fitness graph like this, where 0, 0 is the wild type and 1, 1 is a double mutant, and these arrows always point in the direction of increasing fitness. And so what you see is, as you increase concentration, you move from a situation where the wild type is the fittest to a situation where the double mutant is the fittest, and in between you have a situation where the two single mutants are both fitness peaks. Okay, and this is sort of the key point, that as you change the concentration, this fitness landscape becomes more and more rugged at some intermediate concentration, and at large concentration, it again becomes simple in the sense that only the maximum number of mutations gives you something that can survive. And so this you can sort of numerically generalize to an arbitrary number of mutations. So this is a simulation for 16 mutations. And if you just look at this dashed curve, you see here the number of peaks in the fitness landscape as a function of concentration. So it has a maximum and then it goes down again. And in the inset, you can see the logarithm of the number of peaks scaled by L. So these are landscapes that are very rugged in the sense that they actually have a number of peaks that is exponentially large in L, which is sort of typical of most model fitness landscapes. However, and this is sort of the final and key point, these landscapes are nevertheless highly accessible. So this is sort of different from the landscape of, for example, Dan Weinreich that I showed you in the beginning. And so there's a theorem that one can prove, which is based on the observation that by the way this model is constructed, certain rank orders of fitness landscapes cannot occur. And if you make use of this, then you can show that any peak genotype, that is any genotype that is a local maximum, is accessible from all its sub and supersets. So what does this mean? So this is a schematic for a landscape with two peaks. So if we look at this red genotype here, which is a peak, then this has subsets. So subsets are genotypes that contain a subset of the mutation. So in this case, this would be one subset and this would be one subset. And the all zero genotypes is of course always a subset. And you see that all of these are sort of accessible, right? So this landscape has a curious structure in the sense that it is very rugged, but at the same time, it's actually rather easy to evolve to the peaks. And at any concentration, the fittest type is always accessible from the wild type.
Okay, it's time to wrap up.
Yes, so I'm done. Yes, very good. Just kind of a couple of very general conclusions. So I think in the first part, I think it's one thing that I found important there is that of course, if you really want to do evolutionary predictions, you need to know something about mutation rates. And I think generally, these are actually much more difficult to infer than fitness effects, and also to measure experimentally, of course. In the second case, in the second example, I showed you that we have the simple structure due to the scaling property and the non-epistatic marginal phenotype. So here the question is, how general is this observation? And one of the things that we're starting to do now is to use the model that I presented to look at resistance evolution at varying drug concentrations. And so finally, let me mention again my collaborators, so the postdocs who worked on this project in Cologne, Arjan and his postdocs in Wageningen, and the Edinburgh group. And thank you very much for your attention. I'll be happy to take your questions now. I think you're muted.
Okay, thanks a lot. So I see a first question by Eric. Is there a correlation between the RI and MI, or do they fluctuate independently, either in your model or in the real world?
So we're not in the model as I presented it. We're assuming that the RIs and the MIs are not correlated, except that the RIs are all less than 1 and the MIs are all greater than 1, which per se already induces a certain amount of pleiotropy in the sense that the lower the fitness, the higher the resistance. But you could also, think of making them correlated. Whether they are correlated in the data, I frankly don't know. This is something that one could maybe check.
Okay, thanks. I actually, now you answered me already by saying all the MI are bigger than 1, all the RI are smaller than 1. I was wondering if this fact that you get most local optima at intermediate doses, if that's an effect of that, or if that would also happen if the RI and MI could both be smaller and bigger than 1 and be uncorrelated.
Right. No, I think this is certainly because of that, right? So this means that basically, on average, the more mutations that you have, the more resistant you are, right? Okay. So and your growth rate goes down. So it's clear that there's a trade-off. And I think I didn't, I probably skipped over that too quickly, but there's a kind of quantitative relation between the number of mutations, the typical number of mutations of highly fit genotypes and the concentration, right? So as you increase the concentration, it pays off to have more and more. And this is because of that condition, yes.
Thanks.
Okay, there's a question by Michael Manhart. Do you have more biological explanation for why you think SNPs are generally larger effect than CNVs? I would expect the opposite. CNVs will knock out whole genes or significantly overexpress them.
So one class of SNPs that clearly have a particularly strong effect in our case are those in the TEM1, right? So these are, the activating, we know that there are a couple of activating mutations in the TEM1, and they of course have a very strong and specific effect on the resistance. On the other hand, I would say no, but I think that you see the point is, of course, that you only see the large effect SNPs. Of course, there are lots of SNPs that have small effects, but I think what is more important is that they also have small rates, so you wouldn't actually see them, right? Now, with regard to the CNVs, I think we not all of them are even beneficial, presumably. So for example, one deletion that happens very often, and that was, you know, a big surprise, is the deletion of the TEM1. Okay, so about half of the small populations get rid of the TEM1, and we thought initially that this was beneficial because we thought, you know, there's a cost associated with it. But if we look at it more closely, we see that it's probably neutral. But it just, because of some, you know, because of the way the plasmid is constructed, it happens to be very easy to delete the TEM1. So some of these deletions are presumably neutral and also maybe or even weakly deleterious. This is not so clear.
Okay, is there more questions? If not, we're going to go to the next speaker. And thanks a lot for this fascinating talk. It's super